Okay. Good evening, everybody. We'll just wait a couple of minutes until folks start filtering in. Okay. Let's get started. In the interests of time. So I thank you all to everybody for attending this third Translating Science webinar hosted by the Wound Healing Society Education in collaboration with Wound Healing U. Today our topic is Beyond the Lab to Standard of Care, Regulatory Aspects of Translation, presented by Dr. Carlos Zgheib. If you have any questions throughout this evening's presentation, please use the Q&A box and we'll address as many as possible at the end of the webinar. And as a reminder, this webinar is being recorded and will be available on YouTube in a few days' time. So I am Kath Bogie, I am the chair of the Education Committee, and I will hand you over now to our moderator, Sarah, to introduce herself. Thank you.
Hi, everyone. My name is Sarah Holtman. I am the moderator for this presentation. I'm a registered nurse and the director of clinical education for Wound Care University, and I'm happy to introduce to you Dr. Carlos Zgheib.
Good afternoon. Thank you for the introduction. I would like to thank the Wound Healing Society and the Wound Healing Education Committee for inviting me to speak today. So my name is Carlos Zgheib. I'm an associate professor in the Department of Surgery at the University of Arizona. I have also joint appointments with biomedical engineering and pharmacology. I lead the laboratory for fetal and regenerative biology at the university with Ken Lichty. Quick disclosure, I'm the Chief Scientific Officer of Syria Therapeutics, which is a biotech company that we started a few years ago to bring some of the technologies that we developed on the bench side to the bedside, drugs, including a drug for treating diabetic foot ulcers. To start this talk about the regulation of translation, it's good to start with defining translation. Translational research is the process of moving your research, your ideas, or your discoveries, your drugs from the bench side to the clinical side, to the clinical application, and eventually, hopefully, into a routine medical practice. So the approach from the bench to the bedside emphasizes on how discoveries in the lab must undergo rigorous process before reaching clinical trials. And that can be divided into really two major phases, which we're going to go in depth about, the preclinical phase and the clinical phase. Basically, the preclinical phase, everything before the drug is administered to human, and clinical phase is that stage when the drugs start being tested on patients. Hence, in order to do all this, there is a crucial importance to regulation. Regulatory science is the science of developing and applying tools, standards, and approaches to assess the safety, efficacy, quality, and the performance of drugs. And it is important to have a regulation because it connects the experimental discoveries with laws and guidelines and regulations that are set by certain agencies or authorities, especially here in the US, which is the FDA. So the FDA, and I'm sure as everyone knows, the FDA is the organization that regulates, that really approves new drugs to be put on the market. And the FDA has several centers. The two main centers that are involved in approving drugs are CDER and CBER. CDER is the Center for Drug Evaluation and Research, and the CBER is the Center for Biologics. And in addition to these two centers, the FDA also has centers for food safety. They have the Center for Veterinary Machine, and also the Center for Devices and Radiological Health. To start with, the Center for Drug Evaluation and Research, or in short, CDER, basically this is the center that's going to approve any molecule or any drug that goes into humans. So it regulates human drugs, which are chemical substances manufactured for therapeutic and diagnostic purposes. And here on the right-hand side, that table shows you some of the things that CDER regulate. For example, monoclonal antibodies, cytokines, therapeutic enzymes, hormones, growth factors. I'm going to talk later on in my presentation about one of the growth factors that was approved for the treatment of diabetic foot ulcers, and it was approved through this center, through CDER. So the review process through CDER, they review the application of a new drug application to ensure, again, safety, efficacy, and quality. And here to emphasize, the FDA priority is safety, then efficacy when they're reviewing new drug applications. So CDER has expertise in a wide range of clinical areas, including cardiovascular disease, oncology, neurology, psychiatry. So the other center is the Center for Biologics, Evaluation and Research. And this center regulates the biological products, which are substances derived from living organisms or their byproducts. These include vaccines, blood proteins, even blood cells, and gene therapy, microRNA therapy, other RNA therapy. And CBER reviews these applications. The application is called Biologic License Applications, application, BLA. They review these, and the process is a little bit more complex than an NDA due to the variability of biological products. They have expertise in allergy, immunology, infectious disease, hematology, and also oncology. So when you when you are developing a drug, investigational new drug or IND, we're going to call it a drug for now. It's a new chemical-based drug or can be biological, and it has to go through clinical trial to get FDA approval and be available on the market to treat patients. So in order to do that, you're going to need to submit an IND application to the FDA. And this IND application going to contain a lot of information about your preclinical data before starting the clinical trials. So in order for a drug to go into clinical trials, the FDA has to approve it first, approve the IND application. And the FDA will want to know a lot about your preclinical data, if it showed efficacy, if it showed toxicity in animal models. Also, this application will include manufacturing information, details about the drug stability, the manufacturing, the GLP manufacturing, also clinical protocols and investigators, like detailed protocols, which population of patients you are targeting, what kind, for example, for the application of wound healing, they will want to know what type of wound you are targeting, size of wound, age of patients, how many administrations you're going to give from your drug. So here, this is basically a snapshot of how drugs go from the bench to the bedside, from preclinical to clinical. You start with drug discovery. You find a drug, or you find different drugs that have potential in vitro and in vivo, for example, here to improve wound healing. And you do the preclinical development where you test in different animal models to show efficacy, but also you would do some toxicity studies. Once you have that data, you submit an IND application with all that data before starting the clinical trials. There's also the pre-IND meeting, which is an opportunity to really have a conversation with the FDA and ask them questions about your drug. Basically, here you put all your preclinical data and you ask questions, for example, about your clinical trial design. If your drug is topical, do you need to do toxicity study or systemic toxicity and other things? There are different types of INDs. The two main INDs that are usually submitted for drugs are the investigator-initiated IND or the commercial IND, which usually is submitted by a sponsor, like a company. In addition to these two types of IND, there's a research IND, treatment IND, and an emergency use IND, where this is for drugs that are needed in case of emergency use. The FDA review process. So once you collect all the data, all the information about your drug, your preclinical testing, your efficacy data, and hopefully some toxicity data in your animals, you put that into a package, into an IND package, and you submit it to the FDA. The FDA has 30 days to review and send back comments. Once these comments are answered and the FDA will approve your application, then the next steps is to go through an IRB submission, which can take weeks to months depending on the institutions. If you are at an institution with really fast IRB approval, you can get your protocols approved within 30 days. Other places can take even more than 6 months. Once that approved, then the clinical trials can proceed. This is an overview of where the drugs start and the drug goes on. Initially, you might have multiple, for example, let's say you're trying to activate a receptor that can stimulate wound healing and accelerate wound healing. You would start with so many different candidates. You narrow it down to few candidates, then you work on one. You make it more stable, you make it more available, you make it more have more effectivity. You test in your preclinical models, then you transfer to phase 1, phase 2, phase 3, and eventually approve. Sure, already most of you know the different clinical trials. We have a phase 1, which is mainly a safety trial, where you have a small number of patients and you want to make sure that your drug is safe based on the dose you're given or the dosing regimen you are giving. Phase 2 is also safety and dosing. And usually from phase 1 and phase 2, you still get some efficacy data. And if the drug usually has some efficacy, the drug would proceed to phase 3 clinical trial and then FDA review. If there's no promising potential for the drug from phase 1/phase 2, usually the clinical trial is ended and will not proceed to phase 3. Some drugs do require a phase 4, which is even after the drug is available on the market, it will need to be monitored and have surveillance and collect more clinical data post-approval. Not all drugs are approved in clinical trials, and the reason why, there are multiple reasons why these drugs fail. On the left-hand side here, this pie chart is showing you that the main reason why drugs fail in clinical trials is efficacy. A lot of times, drugs that show really promising effects in animal models, these effects do not translate into humans. Some would say 98% of the drugs in preclinical trials that are effective fail in clinical trials. The second reason why these drugs fail is the safety. They could show significant side effects or toxicity, and the FDA will stop the clinical trials. On the right-hand side, we have the percentage of failure by therapeutic areas. The biggest failure is in oncology. The second biggest area with the failure of drugs is the drugs for the central nervous system. This chart is showing the probability of success of the drugs moving from, for example, from phase 1 to launch, phase 2 to launch, phase 2 to phase 3. So the bottom line is showing you that the rate of success is around 6% for drugs from phase 1 all the way to launch. Phase 2, it's a little bit higher. It's between 14 and 15%. And usually when you reach phase 3, if the drug showed efficacy in phase 1 and phase 2, there's a high chance it's going to show more efficacy also in phase 3, where you have a higher probability of success, hence the increased up to 60% when you reach phase 3 trial. Once you complete the clinical trials and everything went well, the drug has efficacy and has a safe profile, in order to put the drug on the market, the FDA will need a new drug application. And this new drug application is basically 500 to 600 pages of all the information about the drug, from the chemistry to the manufacturing to the stability about the drug, but also pharmacokinetics, toxicity, bioavailability, as well as the results from the clinical trials and patient information. So the NDA submission process, once the NDA package is complete, you have the option to submit this NDA package to do a pre-submission, just like the pre-IND, do a pre-submission to get input and discuss it with the FDA, or you can directly go towards a submission submitting the package to the FDA. The FDA will review the package and give probably comments and questions, and it's going to be back and forth. And eventually, if all questions were answered, the drug will be approved and available on the market. I mentioned early on CBER, which regulates the biologics, and instead of an NDA, we would need to submit a BLA. There are similarities with the NDA, which contains, again, the manufacturing, the clinical and preclinical data, but also there are unique challenges to this is because it's more complex than a chemical drug or a molecule compared to a biologic. So the BLA submission review process is a bit similar, but it includes extra work. There's the initial filing, then in-depth review, and there's inspection of manufacturing facilities. The FDA may conduct inspection of the facilities where you are making your gene therapy or stem cell therapy. Then the findings will go to an advisory committee meeting. This panel is made out of experts that will review the application, will provide valuable insights and recommendations to the agency. The possible outcome, or the hopeful outcome is going to be the approval or the request for additional information, and in rare cases, rejection. In addition to these two paths, there is the orphan drug designation. And the orphan drug designation is for drugs that are developed for rare disease that affect fewer than 200,000 people. The advantage of this this designation compared to the standard NDA application is that you get tax credits, there are fee waivers, and 7-year market exclusivity. This is done by the FDA to really encourage investigators who are working on rare drugs for rare disease. In addition to that track, there is the fast track designation. And from the name, you can tell that the fast track designation, it means you're going to get faster approval for your drug. And this is usually done after phase 2. The FDA will review the information and about the drug and recommend approval. Then phase 4 can continue. This program is to expedite the review of drugs for serious conditions that address unmet medical needs. And usually for wound healing, this fast track designation doesn't apply. It applies in more serious disease. There's also another track for breakthrough therapy designation. It's similar to the fast track, but it's for drugs that show really great improvement over existing therapies. And here the FDA would give significant support even during the development, during the preclinical development, and help with accelerated approval pathway. For example, here, drugs for cancer and rare genetic conditions. The accelerated approval, it allows earlier approval for drugs for serious condition based on surrogate endpoints. For example, if a drug is tested for oncology and it shows a tumor shrinkage or reduced blood markers of a cancer, that would fall under accelerated approval, and the manufacturers must confirm clinical benefit in post-market studies. Priority review, the FDA designation shortens the review time from 10 months to 6 months for drugs offering significant improvement in treatment. Like, for example, if there is a drug right now approved for diabetic foot ulcers and you have a drug that's showing better effect and better safety profile, you might be able to do a priority review with that. So before going into the clinical trials and an IND application, the FDA requires that the drug that's going to be tested in humans is GLP manufactured. And also, it's good to have some toxicology studies before going into clinical trials. And let me go into a bit more details in these two sections. So the GLP, it's a set of principles intended to assure the quality and integrity of your drug product. basically, the drugs that are tested on animals usually are for research purposes only and cannot be used in humans. So the GLP standards, they ensure that the data is reliable and reproducible, and they are required by the FDA for studies submitted in IND applications. The key GLP components are SOPs, all the detailed protocols for manufacturing and testing the drug, documentation, and the quality assurance unit, which is an independent group ensuring the compliance with GLP. All these are governed by the FDA's 21 CFR Part 58 and also under the international harmonization through the OECD GLP principle. Toxicology studies definitely is to show potential adverse effect. And if you are developing a drug on the preclinical side, on the bench side, and you see that it has some efficacy, it's good to do a small or some toxicity studies if you can to make sure the dose you are using, as well as a certain doses that are higher from the dose that's efficacious, do not cause toxicity or significant abnormal pathology, because you don't want to start a clinical trial where a big clinical trial and you don't know, and the drug will show these toxic effects. The types of toxicity studies, there's multiple. I'm not going to go into much details, but there's the acute, subacute, chronic. There's also genotoxicity. This is where the drug has a potential to cause genetic mutation. There is the carcinogenicity, potential to cause cancer, and also effects on fertility and fetal development. Usually these toxicology studies are done in two species, rodent and non-rodent. For wound healing purposes, rats and pigs are the two species that the toxicology studies are done in. And the dose selection, multiple dose levels, as I mentioned, depending on the dose you're starting with in the preclinical studies. This graph is showing you the toxicology studies where you can do them. They are a priority during discovery. You can do small toxicity studies in the preclinical studies, and the phase 1, you do more toxicity study, pharmacokinetics and PK studies. And over this time, you can, during these phases, you can talk with the sponsor and request meetings with the FDA to discuss your findings. Clinical trial and ethical consideration. There are 4 different points here that I'm going to discuss. First, I'm going to start with the clinical trial protocols. It's a detailed plan that describes the objectives, design, methods, the statistical consideration, and organization of the trial. When you are designing a clinical trial, it's crucial to design the clinical trial that's gonna help your drug be effective. For example, you gotta set your primary and secondary goals. If your primary goal improving wound healing, what would be your secondary goal? Improving your wound healing by what percentage? A study design, is it randomized, double-blinded, placebo-controlled? Inclusion and exclusion criteria. Define the patient population that you're gonna test your drug in. How old are these patients? What type of wounds do they have? Diabetic foot ulcers? Do they have venous ulcers? Do they have different wounds? How many wounds do they have? Do they have wounds on both limbs? What's the size of the wound? Are you going to include the patients that have a 4 centimeter square wound and below, or you're going to include bigger wounds? All these criteria would play a big role in the in the success of your clinical trial and in the success of your drug. Also, the statistical method you're going to use, the sample size calculation, data analysis plans. The clinical trials must be reviewed and approved by regulatory agencies and IRBs, and there are opportunities to submit amendments and deviations from these protocols, but these will be regulated by the regulators. So the IRB, the main role of the IRB is to protect the rights, safety, and welfare of human research participants. And again, the priority for the FDA is the safety of the participants, then the efficacy. The IRB responsibilities here are they will review and approve study protocols. They will ensure informed consent is appropriately obtained, and monitor the ongoing studies for compliance with compliance and safety and the design that you included in your IND application. The IRBs have diverse memberships that include scientists and non-scientists and community members, and these are governed by FDA regulations 50, 56, 45, and 46. Informed consent, it's a process by which a participant voluntarily confirms their willingness to participate after being informed of all the aspects relevant to their decision. When recruiting patients, the patients will want to know a lot of information about the trial, and that's when they will want to know the potential risks and benefits. They want to know the purpose of the study. They want to know what medical procedure are they going to go through. They want to make sure their information is protected, where the location is going to be, is it close to where they live, the costs and reimbursements. Some clinical trials offer some incentives to recruit patients and, more importantly, to keep the patients in the clinical trials. Because one of the hurdles, if the clinical trial takes a long time, some patients drop out. That could affect the outcomes of the clinical trial. And all this information must be documented via written consent approved by the IRB, and there will be special, if you want, additional protection for special populations like children, pregnant women, prisoners. These will need extra regulation into the informed consent to make sure they're protected. Finally, the data monitoring and safety. The data monitoring and safety committees, they are independent groups that review the data during a trial to ensure the participant safety. And there are safety monitoring plans to outline procedures for reporting adverse events and protocol deviation. And there's SAEs, the serious adverse event, where patients or as well as sponsors can report any issues directly to the regulatory authorities and IRBs. Risk management. It's good to have Plan B and Plan C, or plans or strategies to minimize risk, such as stopping rules or dose adjustment. If the dose showed significant effect, should we reduce the dose? Is the dose given daily? Should be given every other day? And all these are regulated under the 21 CFR Part 312. So in the US, the FDA is the agency that regulates drug approval and medical device approval in the US. In Europe, it's EMA, the European Medicines Agency, and the agency there have a centralized procedure and decentralized procedure. The centralized procedure is for drugs that are going to be approved to be used all over the European Union. The decentralized is going to be approved by a specific country for the use in that country. The other regulatory authorities, there's the Health Canada that regulates drugs in Canada, PMDA in Japan, and TGA in Australia. Now I want to talk about, I want to show you 2 case studies about, one case study is about a drug, and I think that's the only drug that has been approved for the treatment of diabetic foot ulcers. And this drug is available on the market under the brand name Regranix, and the active compound is becaplirmin. It's approved by the FDA for the treatment of diabetic foot ulcers, and it's basically it contains the human platelet-derived growth factor, which promotes the granulation tissue formation and enhances wound healing by stimulating cell migration. And as I mentioned early on in my talk, since this is considered a biologic, it was submitted as a BLA application to the FDA. It was improved in 1997 after reviewing preclinical, clinical, and manufacturing data. However, there has been some key regulatory challenges with Regranix. One is safety monitoring. Later on, with more research and more investigation, the drug was linked to a potential increased risk of cancer because it promotes cell growth, which required the manufacturer to add a label onto the box about safety. Post-market surveillance, it underwent continuous post-market safety reviews, and new warnings were added based on the safety data. Translation into standard of care. So Regranix by itself is not used by itself. It's used by healthcare providers to manage the FU in conjunction with good wound care practices, such still need to do debridements, still need to clean the wounds, still need to keep the infection under control, in addition to adding Regranix. Limitation, it showed it's effective in specific cases, but it's high cost. I think one tube of Regranix costs like $1,500, and the black box warning have limited its widespread use. But still, it has clinical benefits by accelerating wound healing in diabetic patients and reduce the risk of complications, long-term complications, such as infections and amputations when used appropriately. The case study number 2 is a device approval. This is quickly. There's also the 501(k) pathway. We all know about the negative pressure wound therapy that is used in different types of wound to stimulate healing and help the wound healing. For example, the famous one is the VAC therapy system, but there's also the PICO system, which was approved because it showed a little bit more, it was equivalent, but already had better outcomes than the VAC therapy system. So clinical studies demonstrated that PICO improved healing outcomes by accelerating granulation tissue, reducing wound area. After approval, the PICO system continues to be monitored and efficacy through post-market surveillance. So the PICO leveraged the 510(k) pathway, which reduced the need for a lot of clinical trials or long clinical trials by showing that it's equivalent to already approved device, but it has some better results. The challenges in drug translation, and to start with the common hurdles, the main reason the drugs fail is the safety issues. You might not see the safety issues in animals, but can happen in early clinical trials, unanticipated adverse effects in humans that can end the clinical trials. efficacy challenges. Again, drugs that are effective in animal models, not necessarily are going to show efficacy in humans. Regulatory details. If the application is not complete, that's going to lead to delays in approval from the FDA. Strategies to overcome these challenges is early and ongoing communication with regulators. Here, the pre-IND meeting with the FDA is important. Also, talking to experts in the field and consultants is also crucial. Best practices for researchers navigating the regulatory pathways. One, engaging with regulators early. The benefits is clarify regulatory requirements. Is your drug going to go through CBER, CDER? What needs to be done? Identify potential issues proactively. If your preclinical data good, is your GLP manufacturing good? You can use the pre-IND meeting, discuss plans before submitting your IND, or the breakthrough therapy meetings. Tips, prepare detailed questions, especially questions about the clinical trial design and the toxicity data you have and the distribution data you have, if it's enough. Provide background materials in advance about the drug and document meetings outcomes because you're going to need them later on. The second one is documentation and compliance from day one. It's important to have really good records. They're going to be essential for the regulatory submissions, and it will help facilitate audits and inspections. Key documents are the study reports, detailed findings from your preclinical studies as well as your clinical studies, manufacturing records, batch records, quality testing, and the correspondence with the regulatory agency is also important. Compliance area, again, the GLP manufacturing, the data integrity. Data integrity is essential to ensure that the data is accurate, complete, and verifiable. Collaboration, the third is collaborating with the regulatory experts. In our case, when we started, we had multiple consultants in different sections and different fields. These experts have worked on a lot of clinical trials, a lot of drugs. They have so much input, and they know the FDA, they know the regulations of the FDA. They help you navigate complex regulatory environments, prepare and submit applications. And in order for a drug to be successful from preclinical all the way to clinical, it's always good to have a multi-disciplinary team. You have the scientists, but also taking advantage of the clinical expertise. What can work? What are their observation? And on the clinical side, what could be easy to use? What can be improved? Also, the legal experts. When you're developing this idea, you're gonna need to protect it by IP and patent, and you're gonna need legal experts as well as regulatory specialists. The benefits is improved quality of submission and increased chances of success. Outsourcing options, I already mentioned consultants and CROs for their expertise. So the key takeaways from my talk today is that regulatory science is essential. It's good to understand the different pathways and choose the best pathway for the drug that is being developed. And that can happen with the early and ongoing engagement with the regulators. And the future is dynamic. It's always important to stay informed about evolving regulations, especially in fields related to digital health and gene therapy. If you're working on a gene therapy, regulations are evolving, they're changing. It's important to keep up to date. So next steps for researchers, continue to learn about regulatory requirements and updates, build a multidisciplinary team, emphasize quality and compliance, prioritize data and integrity, and at the end, advocate for patients, focus on delivering drugs that are safe, effective, and importantly, accessible treatments that can make a difference in patient lives. With this, I end my talk. I would like to thank you for listening, and thank you again for the Wound Healing Society and the Education Committee for inviting me to present, and I'm happy to answer your questions.
You can type your questions in the Q&A box, but we'll ask the first one. You said that, and again, my background is nursing, so very different. You said that the main reason for failure is safety issues. What percentage of those would you say go back to the drawing board and try again versus those who just completely stop?
It depends on the drug, the
Okay.
nature of the drug. If it's a chemical drug, you could go back, do some adjustment to the molecule using medicinal chemistry, change certain groups on the molecule. Maybe that could reduce toxicity. Also, sometimes when you start with the molecule and you change it and you put it in clinical trials, it can lead to more toxic effects. When it comes to other type of products that cannot be changed, the best is to choose a different drug or a different molecule and go from there.
Thank you. Any other questions? You can type in the Q&A box.
Okay. I did have one question. You talked about the changes that are going on in the field, and maybe I missed it, but do you have any thoughts about AI or how AI or machine learning are going to impact FDA approvals? You know, do you maybe not need to do all the animal studies?
Exactly. With the AI, there's a significant advancement with AI where you can, at different levels. One level is screening drugs using AI. Like, for example, if you're trying to activate a receptor to enhance, I don't know, stem cell recruitment to promote healing, you can use AI to really figure out which molecule is going to be the closest to activating that receptor instead of trying to screen 2,000, 7,000 different drugs. Probably it can give you few that you can start with. And if you have efficacy, you can continue from there. The other is where the FDA can use AI is using their records on previous drugs and previous molecules that failed in clinical trials and compare new drugs that are being submitted and see the potential of them failing. But also, more importantly, perhaps in the future, AI can tell you if modifying this molecule in a certain way is going to cause more toxicity because there's a history of that group causing toxicity.
So it could be very useful, but there's a lot of questions in development.
Yes.
Still. So thank you very much, both of you, for this presentation, and thank you all for attending the final Translating Science webinar series brought to you by the Wound Healing Society Education Committee and Wound Healing U. As a reminder, the recording of this presentation will be available on the Wound Healing Society YouTube channel and the Wound Healing U website in the next few weeks. This is the last in our 4 series of Education Committee webinars, but we will be back with something new and different in the spring. So keep posted. Right. Thank you, everybody.
Thank you.
Bye.
Thank you.